Hello class, Mr. Allman here. I want to give you today a little bit of an overview into DNA. DNA is a polymer, and as a polymer, it is made up of monomers known as nucleotides. And those nucleotides have really three parts. They have nucleic acid, they have a sugar, and they have a phosphate. When we look a little bit more into the molecular structure of DNA, it has the standard four: carbon, hydrogen, oxygen, and nitrogen. But then it's got this fifth element in a pretty large amount, and that's phosphorus. And we see phosphorus in things like ATP, but we don't really find it much in proteins, and certainly not at all in carbohydrates and fats. So that's one thing that sets DNA apart from other types of molecules in the body. It comes in two flavors, DNA and RNA, and those are fairly similar. If you know the structure of one, you can pretty easily find you can know the structure of the other. They certainly do have different functions, though. So we have the phosphate, we have a pentose sugar, pentose meaning five-sided, so it has a ring of five sides, and then a nitrogenous base. Those bases are those A's, T's, C's, and G's that you might be familiar with from freshman biology. Now, as we look at DNA versus RNA, the difference in the structure is the sugar. DNA has deoxyribose and RNA has ribose. Ribose is the name of the sugar, deoxyribose in RNA. Deoxyribose is simply a ribose sugar that has one less oxygen in it. The bases that they contain, DNA has A, T, C, and G, adenine, thymine, guanine, and cytosine, whereas RNA replaces the thymine with uracil. DNA is usually two-stranded, meaning it's got two strands that are coiled to form a double helix, whereas RNA is usually found as a single strand. To look at DNA in terms of some of its function, two main functions. Number one, it stores information about how to make proteins, and that information that it contains is the sequence of RNA molecules that will be used to determine the sequence of amino acids that's used to build the protein. The second important function of DNA is that it's heritable, so that information that's stored is passed on from one generation to the next through the process of reproduction. Now when we look at the structure, you can see that there are two chains, and those two chains are coiled around each other. Within a single chain, they're covalently bonded to each other. It's known as a phosphodiester bond. It's a covalent bond, know that. So that's what holds the sugar and the phosphates to each other. Now, the nitrogenous bases are linked together. This would be like the rungs of the spiral staircase, and those rungs are going to be held together by hydrogen bonds. And so usually represented here by the dots. And those are a weaker type of bond that is more easily broken. There's a pattern to the way that they are joined together. Adenine binds to thymine, and guanine always binds to cytosine. So A with T and G with C. Here you can see the colors representing the different types of atoms that are found in it. So you can see a nice array, a lot of nitrogen, a lot of carbon, a lot of oxygen, and then again those phosphoruses there in yellow. So it's a fairly diverse molecule in terms of its atomic composition. RNA, in contrast, its functions, it's going to be the go-between from DNA and into the protein, so it's transmitting that information from the DNA, translating it to make the protein. It has a number of other functions in terms of regulating what's happening inside cells, and it exists not just within the cell itself, but there's also forms of RNA found in things like ribosomes and mitochondria and chloroplasts in plants. And so this gives you the overview. It's known as the central dogma of molecular biology. DNA can copy itself. DNA can be turned into RNA, and RNA will be turned into protein. Under special circumstances, RNA can copy itself, and under very particular circumstances, RNA can be turned back into DNA. So that storage of information is what's very significant here. To go a little bit back, you have some of this historical context or some of the other things that you've read and seen for class. But just to give you an idea, this is not something that happened overnight. It wasn't just a few guys who got together and figured out what the structure of DNA was. But there was really decades of work to be able to figure out what the sequence and what that structure of DNA really was. So we have genetics was known for a long period of time. We knew of chromosomes. We could see chromosomes with microscopes when cells divide, and we knew that there was some heritability of traits, traits being passed on from one generation to the next, back into the 1800s. Looking back into the 1920s, we have Frederick Griffith, who noticed that there was a transformation possible, and that we could have information being passed from one bacteria to another, and it was not just transmitted from the reproduction from living bacteria to other living bacteria, but dead bacteria could actually transmit important traits to living bacteria. And then the Hershey and Chase blender experiment, this earned a Nobel Prize, figured out that DNA was specifically the molecule of heredity. It wasn't proteins, it wasn't the other stuff that was in cells, but specifically it was DNA that encased the information that was being transmitted from one generation to the next. Chargaff's rules, in this case in the early 50s, this was important in being able to figure out one of the patterns. What he did was that he did a lot of careful purifying to be able to see the ratio of adenine, guanine, cytosine, and thymine to each other. And no matter what size sample he took, he found that every time, no matter what the species was, what the sample of DNA was, every time that he looked, he found that the amount of adenine and cytosine, the amount of adenine and thymine would be similar to each other, and the amount of cytosine and guanine would be roughly equivalent to each other. Again, you can see not exactly the same, but that's within the within the range of acceptable experimental error. And particularly because he looked at a lot of species and a lot of samples, he saw that the amount of cytosine and guanine was always the same, and so that ended up being a good clue to figure out how it is that this thing was put together, that cytosine and guanine were paired, and that adenine and thymine were paired. Then we bring later into the 50s, and we get this race between a number of people. Linus Pauling, James Watson, Francis Crick, Maurice Wilkins, and Franklin were all big players in trying to figure out what this structure of DNA was. Obviously, Watson and Crick earned the Nobel Prize along with Wilkins for the discovery, and it's certainly Watson and Crick that ultimately gained most of the notoriety for determining the actual structure. It was the X-ray crystallography, which was a fairly new method at the time that unlocked that key piece of information that they needed to be able to figure out the structure. And that obviously was Franklin's doing, and unfortunately for her, she was no longer alive to be able to share in that Nobel Prize, and certainly it was her work that killed her because of the danger at the time of working with X-rays. Now, the one final important piece of information is about DNA and how DNA gets stored within cells. They get stored not as a single piece of DNA, but it gets chopped in each nucleus into parts, and those parts are called chromosomes. And each chromosome itself has tightly packed DNA molecules, each of which has hundreds of millions of base pairs. To go back, if we look on the left here, this shows karyotype. Each one has a different color here, showing a particular dye that's been added just to be able to help us to visualize the 23 sets of chromosomes. Here is the corresponding picture. If the DNA was spread out, we can see different regions of DNA corresponding to the chromosomes that they belong to. Now, when we look here at this image, this shows DNA. Now, DNA first is going to be wrapped like a coil around histones. Histones are a type of protein. Those histones will then clump together, so you have DNA wrapped around a histone, and then the histones wrapped around each other, making these very large loops. Then those loops themselves are going to be looped together around this central protein scaffold to be able to make what goes together to make the chromosome. So you have this DNA wrapped around proteins, proteins wrapped around other proteins, proteins that are looped, and looped again. So you have these multiple layers in which the DNA is going to be wound and wound and wound and packed. And that's because the pure length of DNA is just so great that there's no other way to be able to fit it into the nucleus without really going through some great lengths to be able to get it all folded up. So again, this is the chromosome. The structure of the chromosome is that you have DNA wrapped around histone proteins and then looped together to be able to make that structure. And that's how our body manages and keeps the DNA in the nucleus. So remember, we have DNA and RNA, similar in structure, very different though in function. The phosphorus is significant. The DNA is a polymer. The fact that it's a polymer made up of the sugar, which is ribose or deoxyribose. We have the phosphate, and then we have the bases, adenine, cytosine, guanine, and thymine in DNA, and then replace the thymine with uracil in RNA. That's the basics of RNA and DNA structure.